Hello, welcome to our webinar today about microplastics and estuarine and coastal ecosystems. My name is Bridget John and I will be your moderator. Today's webinar is hosted by the International Association for Impact Assessment, or IAIA. We are the leading global network on best practice for using impact assessment to make informed decisions. Today's webinar is one of a series of webinars offered by IAIA, and I invite you to visit our website and check out the recordings of a few of our recent ones. As you'll see on the slide there, we have quite the variety of webinars. The next slide that's coming will show you that they show gender, emerging technologies, biodiversity, indigenous peoples, psychosocial, health, resettlement, and a variety of others. So please check out our website. The URL is on that sheet. And if you feel like tweeting out today some of the things that you're learning in our webinar, you'll see our Twitter handle there, as well as the hashtag for our webinar series. Just a bit of housekeeping before we get started. You can see either an orange or a red arrow on the top of your go-to webinar control panel, likely on the right side of your screen. And you can use that to click on it to make your control panel fly in or fly out. We are indeed recording the webinar, so you will be receiving a link within a day or two with a link to the recording and to the slides. It's a 60-minute webinar with about 10 to 15 minutes for questions. At any time during the webinar today, please feel free to enter your questions on the gray questions bar that you see in your control panel. If you expand that, you can type in your questions. So don't feel you have to wait till the questions section. I'll be moderating that as we go along, and we'll be posing those questions to Bob when we get there. And as I said, the slides are available. You'll get a link to those in a day or two, but also you can download them now in the gray handouts pane of your control panel. Our presenter today is Bob Murphy. Bob is a fisheries ecologist at Tetra Tech. Previously, he was a director and senior scientist at Ecosystem Solutions and an adjunct professor at Johns Hopkins University. Bob has rich knowledge in ecology, environmental policy, habitat assessment and restoration, and environmental impact assessments. Bob has extensive experience in both freshwater and coastal systems. He recently co-chaired a workshop on the state of microplastic research and knowledge gaps in the Chesapeake Bay, and he authored the resulting technical report, which provided recommendations on the future of microplastic assessment in the Chesapeake Bay watershed. So you can see he's very qualified for this presentation today. So now I'm going to turn the presentation over to you, Bob.
All right, thank you, Bridget. And thank you everyone for tuning in today. I appreciate the opportunity to discuss this issue and where we are and how we understand it, the state of the knowledge. As you can see, there is been recent work. There's been a lot of recent work globally. In addition, I'm going to focus mostly on a regional understandings and use it as a case study. And this will be in the Chesapeake Bay region. So this is the Mid-Atlantic region of the United States. As we move forward here, I would like to acknowledge a couple of goals that I hope to accomplish. The first being to provide an understanding of the scope of the issue of microplastics, particularly in the aquatic environment. That's my area of expertise. There are other ecosystems where it's an issue. I'd like to review a bit of the state of the knowledge, where we are currently in 2020. And again, I want to use the Chesapeake Bay as sort of the framework from a regional understanding perspective. In addition, we'll touch upon some current efforts to assess the role of microplastics in the environment, what potential impacts are, and why we should be concerned. In addition, I will also touch upon some future directions, which will be many, so I'm not going to spend a lot of time, but it is an emerging issue. It's an emerging issue scientifically, so there's quite a lot of research ongoing. Let's get into this. I will point out The two pictures that I have here are pretty common in the microplastics world. If you're doing work in the aquatic environment, the 5 gyres group found plastic-laden stomachs in rainbow runners. That's the fish that we see in the right-hand side of the slide. NOAA, which is the National Oceanographic and Atmospheric Administration in the United States, took this as a micrograph of the filtration apparatus on oysters. American oysters, a common food and valuable for their habitat provision on both coasts of North America and actually globally. And because they're filter feeders, we can see that they ingest microplastics as evidence there. So just these are the things that we're going to talk about today. This may not come as a surprise to most people. Plastics truly are a global problem. The production of the production of plastics in general has grown tremendously over the last 30 to 40 years. A lot of this, as you might see in this is from a paper published in 2017, Science Advances. A large amount of that plastic production goes to packaging. You can see by the blue portion of this graph. Now, I want to use one example from this production, plastic straws. These are commonly used throughout the world. In the United States, it's estimated that every day we use about 500 million straws. That's a lot of straws. But what does that really translate volume-wise? Because when we think about plastic pollution and plastics in the environment, we think about volume, the amount that is out there and getting into the system. So a box of 40 straws, which is very typical if you go to your local grocery store. So Harris Teeter is a local grocery store in the United States. The dimensions of a box of 40 straws is about 576 cubic centimeters. So, and that's 40 straws. So if we do the algebra, 500 million straws, work down through the calculations, that is equivalent to about 7,000 cubic meters of straws consumed per day. Now compare that with the volume of your office or your room or your house or wherever you might be sitting right now. I can speak for myself. I did my calculations this morning. My office is about 75 cubic meters. This is an enormous amount of plastic that's being produced by just one item that's consumed every day in one country. And this is happening globally. So it's a very nice way, if nice is the word, to understand the scope or scale of the issue. Now, this is not a new issue. Macroplastics have been in the news as an environmental problem for several decades now. These pictures, just a random collection of microplastics in the aquatic environment. You can see in the upper left, just floating bags and empty beverage bottles often can trap organisms. The 6-pack rings that hold beverage containers on the right, very commonly known to entrap organisms in the water, fish, seabirds, turtles. In the lower right, we see a sea turtle that's entrapped in derelict fishing gear. So this is another source of marine debris, particularly plastics, into the environment. The middle picture is another sea turtle, and maybe some of our listeners might be familiar with this because this created a lot of newsworthy coverage because that is a straw that's coming out of the nostril of the sea turtle. And this created a lot of stir in the public, which led to some policy changes. So when issues like this become in front of the public eye, policy changes do occur. We're going to touch on that next. Now I'm going to talk just about our local, again, the Washington, D.C. area. So this is the mid-Atlantic. And if you look at the lower map down here, Maryland, the states of Maryland and the state of Virginia flank the city of Washington, D.C. We have the Chesapeake Bay that runs down the eastern spine, the Atlantic Ocean to the right. Washington, D.C. initiated a plastic straw ban in 2019. If anyone might be familiar with The Daily Show, they actually did a story on this. It was humorous, but was actually fairly accurate in some ways. Currently, the state of Maryland in the current legislative session is considering banning single-use plastic bags. So these are the ones that you would have at the grocery store and other consumer uses. The state of Virginia is considering a different approach, but also with the goal to reduce plastic bag consumption, and that would be tax. They're trying to encourage a behavior change through taxation. And just recently, the state of Maryland banned single-use styrofoam food containers, and that ban goes into effect July 1st of this year. So what is it about plastics and microplastics? Unfortunately, the image on the left may be too familiar with people. These are styrofoam and other plastic beverage containers that are just trash now in the environment. And they start off recognizable as their initial use, and they break down over time. Sometimes it's not that long, and turn into what we see on the right are much, much smaller component pieces. And as folks are well aware, the different forms of plastic, the different polymers, they don't actually break down into their chemical constituents. They just break down into smaller and smaller pieces and don't actually biodegrade. But these are what we term microplastics. So it's worth some time just to define what we are talking about. So microplastics, as indicated, are small plastic fragments. So these include fragments from larger pieces. These include fibers, like polyester fibers, for example, from fleece clothing, and granules. Now, how small are we talking about? I'm going to refer a lot to the scientific literature as we go through this discussion, but typically microplastics in the scientific literature varies in size from about 1/10 of a micron to about 10 millimeters in size. Now that's a size range of over 5 orders of magnitude. For most of the purposes from the studies, the cutoff is about 5 millimeters, but that's part of the issue, is that the standardization of size classes still needs to be resolved. Microplastics are generally divided into 2 broad categories: the primary microplastics. So these are manufactured as microplastics, as small pieces. These are found in products such as facial cleansers and cosmetics. These are a source of microbeads. Primary microplastics are also used as vectors for drug delivery. They're often used for air blasting, so to remove rust or paint from a surface, as opposed to sand, plastic. And lastly, the virgin plastic production pellets, commonly referred to as nurdles. These are the virgin plastic from which other materials, other macroplastics are made from, and they're molded into those products. Now, the secondary microplastics these are the pieces that are broken off the larger plastic objects. So this is through physical or biological or chemical processes, usually through the physical process of just tumbling. If it comes out in the environment through litter and it tumbles through a stream or whatnot, it'll break down into smaller and smaller pieces. This has gained a lot of attention because they're showing up everywhere. Microplastics have now been shown to be ubiquitous, and I just wanted to pull a couple of diverse research articles that showed that microplastics are found everywhere. And I apologize that I used 2 examples referring to beer, but this seems to get people very motivated to study the issue and to assess what's going on. It is found in tap water. They are found in sea salts. But more importantly, when we talk about it being a global issue, they're found in very remote, fairly uninhabited regions, which means there is a global transport of microplastics. So, for example, they've been found in the marine sediments in Antarctica, very low human population in Antarctica. There's been atmospheric transport. In the recent study, sampled atmospheric deposition of microplastics in the Pyrenees Mountains in France and found them, which is a very low populated region and it's at a very high altitude. The point being that they are found everywhere. And this research has been ramped up quite a bit over the last decade or so. If you look into publications, this is a proxy, this figure I'm showing here, the Coastal and Estuarine Research Federation, which is a society of coastal scientists, has a biannual meeting. We did a quick look at the presentations that have been given. So these are oral presentations over the last decade. And as you can see here, it wasn't until 2013 that plastics even made it into the research arena at these conferences. And then it spiked this past year, and the blue portion of the histogram shows microplastics. So it really is a very, very hot issue. There's a lack of understanding of what exactly the impacts of microplastics might be. So when I'm talking about the coastal environment, this is estuaries and the marine environment, tidally influenced regions. But where are the plastics coming from? Where are the origins? This is a report that EcoWatch produced in 2016. And the numbers are still, they might be higher now, but the proportions are pretty accurate. And they estimate that this is a very busy slide, and I apologize for that, but the takeaway message is that about 75% of the microplastic and macroplastics, for that matter, in the coastal environment comes from land-based. This shouldn't be surprising because plastics come from human activity, and humans obviously live on land. But that's also something to think about when we think about policy decisions and changing human behavior. This is another schematic. It was from Zhang in 2017, who was looking at the fate and transport of microplastics in the coastal regions. And as you can see, the river input is the primary source in the upper left. And this is where microplastics get incorporated at different points in the aquatic environment. So they can be incorporated into the coastal regions of beaches and wetlands, where they get buried, or they just may be intermixed with the sand, or in the wetland surfaces. They can be fragmented and carried further offshore where they float. They can be ingested through a variety of marine organisms. We'll talk about that in a little bit. And those that are not ingested or do not make their way through the food web make it down to the seabed where they are incorporated into the sediments. And again, the example of the Antarctic is the case across all of the global oceans. They've been found in deep sea sediments. So they truly are ubiquitous. Now, why do we care? I think that might be self-evident, but the Australian Commonwealth, the Scientific Industrial Research Organization, recently estimated, and this was less than a year ago, that 95% of all seabird species will ingest some form of plastic by the year 2050. So that's not that far off. The World Economic Forum projects more plastic in the ocean than fish, which is a very disturbing idea by the year 2050. This picture on the right is a picture taken on the Anacostia River. The Anacostia River, just to give context, you can see my cursor is over. The Anacostia River flows through the eastern portion of Washington, D.C., and as the watershed encompasses the area around Washington, D.C. in the state of Maryland. It's a heavily urbanized tidal system, and as such, and for a variety of reasons, has a fairly large trash load, including lots of plastic debris, as can be seen in this photo. This is probably taken after a recent rain event. So where does that plastic go? That's the big question, and they're getting a better understanding of the transport and fate of microplastics throughout ecosystems. This is a schematic, by no means complete, that shows potential pathways of microplastics through a system, through the aquatic ecosystem. And in this case, we focus on how it might reach a species that is of interest to humans for consumption or for economic reasons. On the east coast of the United States, striped bass are a very important fish species, but economically, they're fished recreationally and they're fished commercially, and they are consumed. So there's a human health aspect to it. However, you have to understand the pathways that microplastics might get to that fish, and that is a process that is poorly understood at this stage. As I mentioned earlier, I am going to use the Chesapeake Bay and its tributaries as sort of the case study here as examples. There is evidence of microplastics in the Chesapeake Bay and within the watershed, so the non-tidal portion as well. Again, we're using the Anacostia River in Washington, D.C. as a very good example of this, potentially an extreme example, and as you can see, the system on the left, the photo on the left, is a typical low gradient stream that has quite a bit of plastic debris and trash in it. And if you were to take those leaf packs, for anyone who has experience doing stream habitat assessment or working streams, you turn over the leaf packs, you can see that there's quite the accumulation of microplastics being trapped there. So, and this is from a trash survey done in Washington, D.C. There have been a few other surveys in the Chesapeake region to look at the microplastic distribution. An organization called Trash Free Maryland did a trash trawl about in 2014, and this is just a surface water trawl. It was across the bay. They had about 30 sites. And they were just screening for the presence and abundance of microplastics. This was a collaboration between the Maryland Department of Natural Resources, that's the DNR, Trash Free Maryland, excuse me, and the University of Toronto. What did they find? Of the 30 sample sites, each site had at least one piece of plastic. And this was throughout the main stem of the Chesapeake Bay, where there's dilution and there's a spreading of it, so you don't have a great urban impact. And they're broken down by types. And you'll see here the spheres, so foam typically represents styrofoam or styrene. Fragments would be fragments that are broken down pieces of larger macroplastics. Fibers are oftentimes the most abundant because those are released through your dryer vents. If you're drying fleece or other synthetic clothing, they are released into the air and then they are deposited in the aquatic environment. I'm going to stay local here to orient you. This, excuse me, this is Washington, D.C. is the square in the lower left of each picture. The Anacostia River is the river that flows through city there and empties into the Potomac River down here at the lower part of Washington, D.C. And they eventually flow into the Chesapeake Bay. One of the county on the eastern portion of Washington, D.C. and Maryland is Prince George's County. And Prince George's County monitors their surface water. So these are streams in non-tidal areas. And part of the data collection includes trash observations. So this is, on the left is actual trash ratings. So the larger the red circle, the greater the amount of trash. And you can see there are hotspots. If you take that map and create it through some GIS software, you can see the hotspots a little bit differently. But as you can see, is we can identify the source areas for trash that enters into the Anacostia system. So what, we ask? Well, where does that trash go? Where are these microplastics likely to accumulate? The tidal Potomac River and Anacostia River in Washington, D.C. supports large, or has the potential to support the vast acreage of submerged aquatic vegetation. We'll talk about this for the next several slides, and we commonly refer to submerged aquatic vegetation as SAV. And this vegetation grows completely underwater, although you can see in this picture it does reach the surface, but it is not considered an emergent plant. There are goals that have been set for the restoration of SAV throughout the Chesapeake Bay watershed, and in DC, in the period between 2009 and 2017, there's a 2,000% increase in the amount of SAV. This is very important because SAV is a critical habitat for a variety of fish, including the DC state fish, American shad. But as this picture might make you think, are these beds potentially trapping microplastics? Could these be a sink? We thought they would be, and it was worth looking into. Background on SAV and why it's important. As I mentioned, it's a very critical habitat in coastal systems. So these are shallow water areas. These are the estuaries, coastal lagoons, and nearshore areas. This is not the deep sea. And these are critical because they provide food and habitat for a variety of species, those which are important directly economically, or those that are forage for those species that are economically important. SAV also physically keeps the water clear. It improves water clarity by absorbing nutrients. Now, nutrients is another global issue that spurs algal blooms. If nutrients can be controlled and absorbed by these vascular plants, we can probably reduce the amount of algal blooms that we see. SAV also physically traps sediments. And this is important. We're going to come back to this. Because this reduces erosion and slows the water. These are photosynthetic organisms, the plants, so they provide oxygen directly to the water column. So a variety of fish, and this picture up here is a blue crab, which is an iconic species for the Atlantic coast, and part of their life cycle relies exclusively on seagrasses. And they seek out these areas for food resources, so the smaller invertebrates that live on the surfaces of the grass bed, on the grass leaves, or they find shelter from predation from larger fish. Underwater grasses are also directly consumed as a source of food for a variety of waterfowl. Now, the issue of the filtration capacity for SAV is what made us think about the ability for them to trap microplastics. We know that they can trap sediments. That's why I say think sediments. They're natural filters. This is an aerial, it's a black and white aerial image taken by the Virginia Institute of Marine Science, known as VIMS in this region, of the Patuxent River in Maryland. This is a tidal river that feeds into the Chesapeake Bay. And this picture is taken during a falling tide, an ebb tide. And what we have delineated here are SAV beds on the southern shoreline. It's a pretty thick and extensive bed that goes up the shoreline. As the tide is falling, so the tide's coming off the wetland in the marsh area, and goes through the SAV beds, you can see there's a clear delineation of clear water that is coming out of the SAV bed as the tide's falling. A very good visual indication of how SAV beds are trapping sediments. That's a good thing. The Chesapeake Bay Program actually acknowledges that one of the benefits that they serve on their ecosystem services is not only oxygen and habitat, but it's to trap sediments and nutrient pollution. Okay. I don't want to make this too data heavy, but a recent paper published by Cassie Herbux et al. In 2017 demonstrated this with some very direct measurements. This is the two circles on the left. These are vectors just showing the speed or velocity of water flow in the upper portion of the Chesapeake Bay. And she was comparing unvegetated areas, so that is A and B, so this is near the channel. And these are the velocities measured 0.3, 0.6, 0.9. And as you can see in the unvegetated areas, water velocity is much greater. Water is moving fast. It's intuitive. On C and D, these measurements are taken in SAV beds. What we should note, this is meters per second, by the way. So the scale changes. Over on the left, we had 0.3, we start at 0.1. So already the scale is much reduced. In the low biomass SAV bed, it's about a third of what it is in the adjacent unvegetated area. So the SAV bed is very much slowing down the water. And where you have a very high density, high biomass SAV bed, water virtually is at a standstill. It starts to move very slowly. And it's because of this physical impact on the way water moves that sediment particles or anything else in the water column falls out because the velocity slows. So we really are interested in how that worked. Okay. So we're thinking that these SAV beds are trapping microplastics in seagrasses. Has there been any evidence that supports this? The first paper that came out that first evaluated this is very recent, just came out in 2018. And they were looking at the surface of seagrass beds off the coast of Belize, and they found, Goss et al. Found that microplastics were accumulating on the leaf surfaces and then were being encrusted by the natural epibiotic community. So crystalline algae, hydroids, things like that that will grow naturally on seagrass blades were also trapping the microplastics on the blades. So there was evidence to support this idea. So we conducted a pilot study in the Potomac River and the Washington, D.C. region. And it was very simple design where we were just taking a sample of plants in the water column by dropping a net over them and harvesting everything that was in the net. And we also sampled the adjacent unvegetated area. So there's a very direct comparison of whether or not the canopies of these grass beds were indeed trapping microplastics. And the protocols are listed there. The results were pretty straightforward. That there, in the vegetated areas, there was a significantly higher number of particles by volume of microplastics than in the adjacent unvegetated areas. So our hypothesis was confirmed. These microplastic types were broken down into the styrene foams, those are easily identifiable. Fibers made up a large proportion, particularly in the vegetated areas, and then miscellaneous. And miscellaneous were not identified the polymer type, just noted by the different colors. Identifying the polymer type requires the use of some very specialized laboratory instrumentation, which we have not done. In addition to SAV as a habitat in coastal areas, oyster reefs are also very important in many coasts in the world. And in our case, very important in the Chesapeake Bay. My colleague, Christine Naus at the University of Maryland, is working on oyster microplastic interactions as part of her dissertation work at the University of Maryland. And I've been grateful that she shared some of these images with me. And for those who are unaware of the role that oysters play in coastal systems, they're very important ecologically because as the reefs grow, oysters have a remarkable ability to filter large amounts of water, clearing the water of not just algae which they're feeding on, but they can also remove sediments, which they then deposit as pseudofeces, as pellets into the sediment. Again, if we're looking at filter feeders and microplastics are in the water column, there's an interest in, are they filtering out microplastics? The life cycle of the oyster is shown here, and there's a portion of the life cycle that is pelagic. It swims in the water column until they find a suitable substrate and they set. So this is a critical area when they are a veliger and pedoveliger, because this is when they are most vulnerable to predation. They are in the water column. They're free swimming. Christine was looking at how the veliger larvae feed on algae when exposed to microplastics. These are electron micrographs showing that they are ingesting microplastics and that it is affecting the way they assimilate carbon from the algae that they're filtering. Other work by Sotherelu et al. In 2016 showed that oyster larvae exposed to microplastics in the water column were delayed settlement, so by up to 6 days. So typically, the veliger larvae are in the water column for about 2 weeks before they settle, that they're exposed to potential predators. In this instance, you're increasing that amount by 6 days, therefore increasing your predation potential. So that could have a serious impact on oyster population potentially. So what are the implications from both of these? SAV and oyster reefs, pardon me, I missed an E in that, serve as critical coastal habitat. They're the two primary subsurface habitats in coastal areas in this area. And they may be compromised. They may be compromised in the ecosystem services they provide. We saw there's a reduction in oyster recruitment. So that is how many oysters recruit to the adult population. If they can't settle, then they are going to be potentially preyed upon. So you have reduced numbers settling. There's also the implications that there's chemical adherence. So this is where some chemicals, PCBs, benzenes, others, may selectively adhere to microplastic particles, which can cause direct mortality or condition effects. What are condition effects? This may be where it might be sublethal, where they are not outright killing an organism, but they will reduce its reproductive capacity. So whereas an organism may produce 100,000 eggs or gametes, it might be reduced to half that, to 50,000. And such reductions are very important. It can have a huge impact on the population level as it moves forward. Lastly, one of the implications for all this, of course, is the human health impacts. We don't know a lot about how microplastics are bioaccumulating through the food web and how that might impact human health through direct consumption. So there are a lot of unknowns, but the implications are pretty straightforward. I want to give a little background on the Chesapeake Bay for those unfamiliar. The Chesapeake Bay is the largest estuary in North America. It's an economic engine for the region. Major cities on the Chesapeake Bay include Baltimore, Maryland, Washington, D.C., Richmond, Virginia, and the Hampton Roads region of Norfolk, Virginia as well. So there's shipping interests, there are, and more importantly, there's recreational and seafood economic interests. The watershed is very large, over 165,000 square kilometers. It's a lot of shoreline, over 18,000 kilometers. There are 150 major rivers and streams that feed the Chesapeake Bay. And then this last bullet might be the most important when it comes to plastic pollution. There's over 18 million residents. A lot of people. Due largely to the fact that Chesapeake Bay is home to Washington, D.C., which is the seat of the federal government, it received a lot of attention in the late 1970s and the early 1980s as the health of the bay declined. This led to the formation of the Chesapeake Bay Program, and for those unfamiliar, it's a regional federal-state partnership. So the states, the federal government, local governments, it's a voluntary partnership that has adopted approaches to conserve and restore the Chesapeake Bay, its ecological function. The Bay Program helps set policy, although they are non-regulatory in nature, and it sets restoration goals. For example, the image here is Jeremy Testa et al., published in 2017, looks at the forecasting, these Chesapeake Bay Program data to forecast the extent of low oxygen events in the Chesapeake Bay. Well, because of these low oxygen events, the Chesapeake Bay Program has set goals to reduce nitrogen and phosphorus. So, and these have been very successful thus far. I mentioned earlier, one of the goals set was SAV restoration. We've seen that in some regions of the Bay. We have not seen it in others. In the Chesapeake region, urbanization is one of the largest threats. This image comes from Bob Worthdow, 2017. On the left, showing the expansion of developed lands just over the period of time, from 1974 to 2012. So the maroon colors, the darker they are, show the greatest extent. And you can see in the regions around the major cities, so Washington, D.C., Richmond, they're sprawled. The populations are growing outside, and land is being converted from forest or agriculture, for that matter, to impervious. And that's where it's supporting the 18 million plus residents. There's a loss of habitat because of this, forest habitat, riparian habitat along stream corridors, loss of wetlands, things that would naturally filter stormwater before it reached the main rivers and the Chesapeake Bay itself. And of course, trash. Trash is what we're here to talk about today mostly. And with the increase in development and increase in population, we're going to have an increase in trash. So how's the Bay? It's important to know how the Chesapeake Bay Program is structured, and this, for a lot of people, it actually takes several years to understand the structure of the Chesapeake Bay Program. It's led by the Executive Council, which is political leadership at the top, but it has several advisory committees, and the Chesapeake Bay Program, it's not unique, but it's certainly led the development of policy development through good science. So one of the advisory committees is the Scientific and Technical Advisory Committee. The Scientific and Technical Advisory Committee does exactly that. It advises, it leads studies, it makes recommendations, and it's based on sound science. Why is this important to what we're talking about? This is where it comes to the regional aspect, and this is sort of a case study where after we started looking at this issue in the DC region, several of us decided to convene a workshop, a 2-day workshop back in 2019 to identify what were the gaps, what's the state of the knowledge, what do we know about microplastics distribution, impact, and policy implications in the Chesapeake Bay. And STAC, the Scientific Technical Advisory Committee, supported this workshop financially. Actually, the report is available here. We did generate a final report out of that workshop. And part of that report and part of the charge that we were given was to develop a series of recommendations. Apologies for the large amount of text on this slide, but it's important to know what came out of that. So we convened over 50 experts and facilitated a series of discussions. I'm going to go through these briefly here, but the first one would be to actually create a team dedicated to plastic pollution under the umbrella of the Chesapeake Bay Program, and to address some of the growing threats as we are understanding more and more. One of the teams, the STAR team, the Scientific Technical Assessment and Reporting, should be incorporated in the development of an ecological risk assessment. Now, I haven't talked a lot about risk assessments, but this is the underlying framework for understanding what the threat of microplastics are to the ecosystem. We also made the recommendation that, this goes back to what I had said earlier in the presentation about STAC undertaking a technical review of the terminology. There is some disparity in the definition of what a microplastic is. If you get really small, we start talking about what are called nanoplastics, which I am not going to discuss here because it has its own suite of issues and science. The fourth recommendation is to develop a source reduction strategy. The Bay Program can do that, and they can develop those that can be implemented at various governmental levels. And the fifth recommendation is to develop a monitoring program. The Chesapeake Bay Program administers water quality monitoring program for probably the past 40, 30 some years, mid 80s. And that's how we understand what's happening in terms of nutrient loads and oxygen, low oxygen events, is through our monitoring. So in order to get a handle on the distribution of microplastics and potential hotspots, the monitoring program could shed a lot of light on that. So those were our 5 recommendations. We also were considering these ecological impacts. So this goes back to risk assessment. We were looking at these long-term environmental stressors. So I mentioned earlier about a reduction in physical condition. There could be increased mortality for those who do toxicological studies. What are the thresholds to kill, to see a mortality event in various organisms? There's delayed ontogenetic development. So this is the example with the oysters that do not settle. Their settlement is delayed by up to a week. One of the other aspects, its potential, is the trophic transfer. So this is as organisms feed on other organisms who have ingested microplastics. If there are chemical contaminants that have adhered to those microplastics, are these being biomagnified potentially? Another thing that has been demonstrated recently is the increased rates of infection from the Vibrio virus. Vibrio is a virus that occurs in aquatic systems. It typically affects oysters and can be deleterious for when humans consume infected oysters. Vibrio apparently has an affinity for microplastics. So that's something else to consider. So we return back to this conceptual model of a risk assessment. Again, this is very simplified. A typical ecological risk assessment could be much more involved. But again, this is a conceptual model that gets us thinking about what do we know and what do we don't know. And a lot of what we don't know are represented by the arrows. We don't know how microplastics may be getting into benthic burrowers, for example, in the sediments. Some we do, some we don't. And so in order to really understand what might be happening at the endpoint, striped bass, we should be following these pathways, seeing what the strength of those transfers might be. So I just mentioned some of our future efforts as I wrap up. There's a lot more to do to understand just the habitats, and I've been focusing on myself has been the submerged aquatic vegetation beds. So we want to further evaluate these. Our intent this year in our research group is to start looking at a variety of fish species, different trophic levels, to see and age classes, to see how much they're ingesting microplastics. And then the development of a full ecological risk assessment. And what we're looking at potentially doing is using the Potomac River, which is a major sub-estuary and tributary to the Chesapeake Bay, as a microcosm for the Chesapeake Bay writ large. I like oysters. I'd like to continue to see them on my dinner plate. I like blue crabs, same goes. I like to fish. All of these I'd kind of like to see remain fairly healthy population-wise. And so I think this is a pretty critical issue. I've been focusing largely on the Mid-Atlantic region of the United States, but this talk could probably cover any region worldwide because it is indeed a global issue. I definitely want to give a couple thanks to people who've been critical to the work we've been doing thus far. I want to thank Matt Robinson from DC's Department of Energy and Environment, who helped put together and co-chaired the workshop with me, and has been very a good collaborator on this effort. Brooke Landry from the Maryland Department of Natural Resources also helps with the workshop and other work with the SAV research. My colleagues here at Tetra Tech who helped field work and developing lab protocols, John Roberts, Chris Wharton, Chad Barber, Jerry Diamond, and Kelly Summers from the United States Environmental Protection Agency, who was also a critical player as we develop the workshop and as we move forward with the development of ecological risk assessment. And with that, I'm ready to take some questions.
Excellent. Well, thank you very much, Bob, for an awesome presentation. We do have some questions and even more are coming in. So please, just a reminder, you can type in your questions in the gray questions pane. That's part of your webinar control panel. So Bob, our first question asks, in countries where sanitation is an issue and packaged water in sachets or bottles are the order of the day, what options are there to reduce microplastics? How can recovery of plastic into energy be improved? Any thoughts on that?
That's a good question. I don't know how well I can speak to the second part by recovery of plastic into energy. But in terms of the first part of that question, and it's a good question, is sachets are very much an issue in certain parts of the world, and they are not recycled, or largely not recycled. And that's, I think, recycling programs can be very helpful in the reduction of plastic entering the environment. But that's just one mode. The other is to potentially cease the production of plastic. I want to say altogether, because that's not going to happen. But it should be, it ought to be reduced. And source reduction is a challenge, and it's a little bit beyond my area of expertise. But from those who I have talked to, particularly on the policy side, the source reduction is the primary way to reduce plastic entering the environment. There's really no better way, more effective way. Again, I mentioned recycling can reduce it, but you're not getting rid of the plastic to begin with. I imagine when they are talking about the converting to energy is probably burning it, combustion. I think that comes with the other wealth of problems for other contaminants released into the atmosphere. But again, I don't know enough about that to speak to it very well. I'm sorry, I can't give a cleaner answer.
All right. Our next question comes from Luis, and he's asking if you had analyzed any other organs besides the stomach, any other tissues in your studies.
No, we have not. We're looking at stomachs in the initial part of the digestive tract only at this point. However, there are several species of fish, and certainly all your bivalves that are filter feeders. So in fish, there has been a few studies that have looked at the gills, since they are feeding or filtering, excuse me, that the plastics can get attached to. As I mentioned, for the bivalves, it's very important to look at the filtration apparatus, because that's where they're going to be collected. We have not looked into the liver. The liver would be where a lot of the contaminants that would adhere to the microplastics would be processed, but we have not done that, and I'm not aware of any long-term studies that have looked at that. So it's a great question, and it sort of sheds some light on there's a lot of unknowns. The data gaps are very large.
Speaking of unknowns, Arundhati is asking about the bacteria that can eat plastics that exists. Is this an option that could be practically implemented in your study area?
Well, that's an interesting question. We're talking these coastal systems, they're open systems. So this is not a pond or a lake, it's a closed system where you control it a little bit more. If these bacteria are not native, or if they have other potential side effects or unintended consequences, I might be reluctant, but that would be an interesting thing to at least start evaluating in a laboratory setting where you can control the population, you can control whether or not they're released into the environment.
All right, the next question's from Manuel. Would plastic in landfills be prone to degrade to microplastics and enter groundwater?
They certainly could be. But in landfills, the way microplastics are breaking down are through abrasion and physical reworking. And in the landfill, that process is essentially stopped because you've buried them and they've stopped. Now, that's not to say that certain types of plastics, in this case, I'm thinking of microfibers from polyester clothing, could possibly be um, uh, through rainwater and through saturation be driven down into the groundwater. They would have to be very small because at that point, almost to the nano size, so a micron or so, in order to get through um, the sediments, the soils. But potentially I could see that being a source.
All right, Suze is asking that mentions that implications are wide, especially for filter feeders, perhaps, but also mentions that there's some research showing that there aren't impacts on these organisms. So can that contradictory research make it more difficult to get action on the ground?
Oh, that's a great question, and that can enter into a very long-term scientific debate. Yes, there has been some work that has demonstrated that plastic particles may be inert, and they can go through they can be ingested and pass through the digestive tract with no effects whatsoever, and that has been shown. Conversely, it also has been shown, as in the case of oysters, that there is an impact. So it depends, I guess, on the impact we're talking about. So reduced settlement from a population ecologist perspective and fisheries that I come from, that's a potentially very large impact. If an oyster, an adult oyster, is filtering and it's not affecting it or killing it, and the plastic is released through a fecal pellet, and it's not having it, well, that's true. It does not have an impact, but the impact is at a different life stage and can have a population level effect. So my answer is it depends, because there are multiple sides to that question for sure.
All right, and we have time for one last question. I apologize we're not able to get to all of them, but thank you very much for sending them in. We really appreciate all of the interaction here. So our last question is from Sarah, and we've talked a little bit about eliminating things at the source. And so she says that given an estimated 46% of plastics in the ocean is comprised of fishing gear, which does break down into microplastics, what types of policies are being implemented against industry in addition to measures being implemented at the consumer level, like banning single-use plastic?
I confess I'm not well versed on how the fishing, when she was asking about industry, I'm assuming it's the fishing industry from the fishing gear. I don't know exactly what is being done to reduce those. I know there are certain measures being taken in the United States on certain fishing gears, so not to get lost. But it's not the same reason or rationale for the reduction in plastic pollution. Those are typically do more because when gear is lost, they continue to fish indiscriminately and catch and kill things. So it's a little bit of a different goal in that case. So I can't really answer what's being done on the industry side to reduce that, other than, I could make some suggestions there are products that can replace plastics. We've known about that for a long time. I will say the plastic industry is very powerful. I would not like to be disrupted, but changing manufacturing practices would be probably one of the ways to go.
Excellent. Thank you, Bob. Thank you, all the participants, for your questions. We're going to wrap up this webinar. We just want to remind you again that you can check out our past webinar recordings on our website. We have a lot of other training opportunities to offer as well, a professional development program that allows for some one-on-one mentoring. Our annual conference is coming up in Seville in May, and so you could check that out on our website. We have symposia throughout periodically, and we have a lot of publications and resources on our website as well. So as you leave our webinar, I just want to say thanks again, Bob, and thanks to you as participants. When you leave, you will be asked to complete a brief survey. Please take just a moment to complete that. It really helps us to improve our future offerings to you. And in a day or two, you will receive a link to the recording and to the handouts, so you can watch for that in your email box. So a final thank you, and we know that your time is valuable, and we hope this was valuable to you as well. See you next time.